So there is here something I want to discuss. It's related to what Yoweri Kaguta Museveni, the president of Uganda, said. So this I'll section it to the politics, the genetics, the physics and optics, and then the neurobiology of blue South Sudanese. Museveni said this in the year 2025 that South Sudanese are not actually black, they are blue. So is it true or false? So I want to build my case. I have some of the premises that I want to rely on it and make a conclusion that Museveni is actually right by stating that South Sudanese are they have a tinge of blue coloration. It's not actually that they are blue, but they have a tinge of blue coloration, and this is actually based on science and theories all the way from 1600, 1802, 1980, and then the claims made in 2025. So let's get into it. So this is what Museveni said. This is what Museveni said. This South Sudanese. This is an image I got in the internet. And Museveni stated here categorically that when you look at the South Sudanese, they are not black. They are actually blue. If you look at them carefully, it's not black, it's blue. This is the verbatim of Yoweri Kaguta Museveni that was stated in a gathering in this year 2025. So is this factual? Yes, there's some element of truth and veracity in this statement. And I want to, based on scientific argument, based on scientific theories that I had stated earlier. So let's move now to the science, and I'll go to the genetics because I believe everything always go to the genetics. Be it diseases, be it anything, we have to go to the genetics. So this is the genetics. Now the genetics, it's a discovery of not so long ago. This is 1980. 1980. This is like how many years? 40 or 50, 40. Should be 40 something. 40 something. By Jeremy Nathan, David Thomas, and Peter Dibb. They're the one that discovered the genetic basis of color vision. So remember, color vision is usually done by the cones. We have cones and we have rods. These are the photoreceptors. Rods and cones, they are the photoreceptors. Rods, it's usually for night vision, what we call scotopic vision. And then cones are usually for day vision, or also what you can call photopic vision. So if you look at this cone, you can see we have this segment here. This is what we call the synaptic terminal. This one usually synapses. It synapses with bipolar cells. Of course, we have the amacrine and horizontal cells. So the bipolar cells, they're in between the photoreceptors and the ganglion cells. Now, the ganglion cells are the one, of course, that will end up forming the optic nerve, which go all the way to the brain for the perception. So this is the synaptic zone, where we usually have mostly glutamate being released. Now, we have this is the nucleus, which is in the inner segment. So this nucleus, of course, it has chromosomes. The 22 pair plus the sex chromosomes. So those are like 23 pairs. So we have all the chromosomes here. Now, there are two specific chromosomes that I will talk about. We have the X chromosome and chromosome 7. Now, the X chromosome, remember chromosome, of course, has a DNA. It has a DNA that are wound around the histone proteins. Then this DNA, of course, it has genes. And then genes usually undergo the genetic expression to give the pre-mRNA that undergo post-transcription modification to give the RNA, the messenger RNA. This messenger RNA, of course, is the one that goes to the cytoplasm, and then with the assistance of ribosomal RNA and tRNA, they can sequence amino acid to form peptides. Now, this X chromosome has specific location that has this gene, the OPN-LW, that's opsin protein, that is responsive to long wavelength. This is usually red. Red has a long wavelength. So this one specifically calls for this protein. Then the same X chromosome, and surprisingly closer there, very close to the one that called for red opsin, we have the one that calls for green opsin. This one is for medium wavelength. So if you compare this one and this one has a medium wavelength. So if maybe I can try and do some drawing, let me use a pen. I don't know if it will work. Hopefully it will work. So it's working. So I want to just illustrate the wavelength. So for the red, I'll draw like this. This is an amplitude. And then I take it further. And then I draw like that. And then this is a wave of red light. So if this is a wave, the wavelength is between these crests. So we have this point here and this point here. So the distance between these two, this is the long wavelength we are stating. So this one is usually given by lambda if I'm still remembering my little physics. So that's the wavelength. You can see that's a long wavelength. So that one is coded by X chromosome. And then after forming this protein, usually it's inserted. Of course, it's packaged within the vesicles. That should be the Golgi bodies. And then it's trafficked all the way to the discs of the cones and then inserted there. Remember, this protein, it's an integral protein that is inserted on the membrane of these photoreceptors within the discs in the outer segment. So an integral protein that spans this cell 7 times. You can see this is spanning. It spanned the cell membrane of the photoreceptor 7 times. That means it's actually a G protein coupled receptor or a serpentine receptor. So this is actually now the receptor for what? The red light. Then the same X chromosome also codes for green. Let me take green here. So it also called for green, but green is now medium wavelength. So we draw the same diagram, but now the separation of this is not very far. Not very far, like that. So the distance between these, this is the green light, it has a medium wavelength. This distance between the crest of this amplitude is where we have the medium wavelength. So that's a medium wavelength. If you compare this wavelength and this one it's medium. It's a little bit short compared to that. So it also called for that protein, and then it's packaged into the vesicles and then trafficked all the way to the discs, and then it's inserted there. So that one will be responding to the green light. And then now, these two, remember, they are on the X chromosome. And the X chromosome, it's actually a sex chromosome. This one, of course, usually undergo remember the meiosis. And in meiosis, we usually have the crossover. So during crossover, there is usually a lot of errors, recombination error that can occur, specifically deletion. So let's say if there's a deletion for this gene, or there's some issues with formation of this opsin for long wavelength, then this will not be there, and this will bring about the color blindness, the so-called color blindness of the red color. So if these undergo deletion, we usually refer to it as there's some name. I'm going into the extent that I never wanted, but let me state. We call it protanopia. Protanopia. Difficulty in saying red. Red is seen as black. And if this gene undergo deletion or formation of a poor protein, this integral protein, then this one is usually known as a deuteranopia. That's difficulty in seeing green color, or color blindness of green color. Remember, they are on the X chromosome, so women are safe since they have two X chromosomes. So one can have a deranged gene, but the other gene, it's okay, so they are kind of a buffer. But for men, we have XY. So if one has an error, then it means that they have the disease. That's why color blindness is so common in men, because it's X recessive. Since we have one X, if it has a deletion, then that's a problem. Now, chromosome 7 is the one now that code for this protein, the opsin, for short wavelength. These are blue. So if I place here, I can draw the blue. The blue, it's this one, it's red. I needed blue. So blue, this one has a shorter wavelength. So this one, I should place it so close. This is a short wavelength. Like that. So this distance, you can see it's very short. So for this one, since it's very short, remember, the shorter the wavelength, the stronger the energy of this light, these electromagnetic waves. So if it has short wavelength, then it means it's very strong. So this one, it's the weakest, then followed by this is the strongest because of the short wavelength. So this is something that the genetic basis of these cones was done by Jeremy Nathan, David Thomas, and Peter Debe. Of course, this from USA, John Hopkins Hospital. It's one that did these discoveries. This is 1980, just the other day. Now, having known that we have these three cones, the red cone, green cone, and the blue cone, now we can actually get to the physics. Let's see what Isaac Newton did. So Isaac Newton, in the year 1600, this is long time ago, discovered that the light that's usually emitted is usually composite. And if you pass it through a glass prism, you can actually separate the lights that are made up of this white light. So it's a composite. The sun usually emits a light that has composite colors, and this is actually the basis of a rainbow. If we have a water bubble, this water bubble can actually act as a prism, and then it separates that light. That's why you can see the rainbow. Now in this case, you can see these colors. We have the red there, we have our green there, and we have our blue. And the basis of separation is because of the wavelength. So we've said red has a long wavelength. So because it has less energy, you can see it's diffracted. They're being separated at different points because of their wavelength and the energies that they have. The blue having short wavelength, it's actually very strong, and you can see where it's being separated at the point at which it's being separated. So these are the lights that are made up of sun. And remember, for us to see, the sun has to hit an object, then that object has to reflect and hit our cones. So we usually see reflection. That's why at night we cannot see colors, because we have to see the light has to hit the object reflect it to our cones, and then the cones now do the neurobiology of color vision. So this is Isaac Newton. So we want to combine the Isaac Newton and the genetics that we've just seen in the previous slide. So let's move on now. This is now how we see. Now this is the sun, which release the light. Light is made up of composite colors, and because they have different wavelength, they are separated at different points. So this light, since it's a composite color, it will hit the South Sudanese. Then South Sudanese, because of the complexion, they will absorb, remember black usually absorb most of the colors. So South Sudanese are actually, they're actually black. They're black. They're black. And because of their complexion, they absorb most of these colors. Remember, we say the sun releases light that is made up of composite colors with different wavelength. So we say the blue has the short wavelength and it has more energy than red and green. So since it has a lot of energy, not all it will be absorbed. Some will be reflected, but most, because of the complexion of South Sudanese, the melanocyte, the melanin, it absorbs most of these rays, and then very little is reflected. But the blue being strong, it will be reflected more than the other colors. So this that is reflected is what actually will stimulate the cones. Since it's blue, now we can see if we move here, now we have our cones that we saw in the genetics. So since the green, most of the green will be absorbed, it will not reach the green cone to stimulate this color. We have the red, it has long wavelength, so it's so weak. It has, the energy is not as much. So most of it will be absorbed. Nothing will be reflected. But blue is the strongest. So it will hit the South Sudanese. They'll absorb some, but not all. And then they reflect. And whatever it's reflected from the skin of the South Sudanese, it's what it will go and stimulate the blue cone. Then the blue cone, of course, we've said it's connected to what? Bipolar cells. And then the bipolar with the ganglion cells. And then ganglion cells forms the optic nerve. Then this optic nerve, it will take to the brain, and there will be a tinge of blue coloration. So this is the theory we call the Young-Helmholtz theory, the trichromatic theory of color vision. That if let's say we had an object that reflected all these light, the blue, the green, and the red, and then all these cones were stimulated, then we will end up seeing the white. So white simply means that we are stimulating all the cones. Since we are recombining the composite that had been scattered. And then let's say if all these were absorbed and nothing was reflected, then we'll see black. So black simply means absence of stimulation of any of the cones. And then let's say if it's red that is being reflected, and then it's only red cone that is stimulated, then we see color red. So Museveni had a point that South Sudanese are actually blue and not black, but it's not that it's on their complexion. They're actually black, or they have dark complexion, but the dark complexion doesn't absorb all the blue color, because it has a short wavelength, so it's strong. Some will be reflected. So the tinge of blue coloration is due to reflection of this blue ray that stimulates the blue cone, and then we end up seeing the color blue. That's according to young Helmholtz theory, or the trichromatic theory that was formulated in the year 1802. So it's actually the point. Museveni was partially right that they are blue, but this blue is not in their color. It's because they can't absorb all the blue color, but they're actually dark. So it's because of stimulation of blue cone that makes South Sudanese appear with a tinge of blue coloration, not because that they have the blue coloration on their skin. So that is it. Thank you, guys. That was my observation. Thank you.